All right, so we heard about cute snails, now it's about cute fossil wasps. I'm Alexandra Viertler. I am a PhD student from the University of Bern in Switzerland, and I work at the Natural History Museum in Basel. And I just finished the first year of my PhD, and I will show you today what I did so far. I'll talk about fossil Darwin wasps. Ichneumonidae, the Darwin wasp, is just the common name that they received recently, I think 2 years or 3 years ago. So I'm going to use their common name. Before I go to the fossils, I would like to say a few things about them in general. Darwin wasps belong to the Hymenoptera, and they are very diverse. There are more than 25,000 species described worldwide. So they're basically everywhere. And they are divided into 42 different subfamilies. And they don't vary only in color or color pattern, as you see in those 3 pictures, but also in size. We have wasps that are 2 millimeters tiny, and we have 5-6 centimeters long wasps as well. And you see here the left picture, you have a very long ovipositor. The ovipositor is some kind of stinger for laying eggs. So with that, they lay eggs on or inside a host. So that's why they are ecto or endoparasitoid. They're killing the host by finishing their development. And they do that. Their hosts are mainly insects, other insects, but more rarely even spiders, as you see here. And they're adapted to the host. So this wasp has very long legs, for example. What's also quite interesting in those wasps is that they're very poorly studied. There's only a single gene phylogeny so far. Why is it not working? So, okay, you see here. So there's quite a mess. A lot of that is not really disentangled, and the relationships between the subfamilies are not so clear. But one of our collaborators is now working on a UC dataset, trying to do a phylogeny that sheds more light into this. My PhD project, what I'm actually trying to do is I want to assess the impact of the last big mass extinction event on those wasps. The KPG mass extinction event was 66 million years ago. That's also the event when the last non-avian dinosaurs went extinct. And it's interesting because it was long thought that this event had no impact on Hymenoptera in general. You see here the orange bar indicating this event, and the Hymenoptera on the right side. There's not much change during this event. But also this was based on family level. And more recently, they found traces that there were signs of extinctions in some subfamilies. So that's what I will try to find out in Darwin wasps. So at the moment, I'm trying to decipher the fossil record, which is quite interesting because, as I said, it's 25,000 species described, extant ones, but only around 270 fossil species, and 3/4 of them are described by only 8 authors. So there's quite a lot to do. This brings me to the fossils I will present you today. They are from the Fore Formation around 55 million years ago. This is a picture of the formation site in Denmark. And I try to taxonomically place those 3 wasps today with you. For that, I did some interpretative drawings of the wasps, visible here. And I gave them some letters here. So we speak of fossil H, M, and L. And the thing is, I wanted to try to assign them to the subfamilies of the 42 subfamilies there are. And because it would be quite a large data set, I just pre-chose subfamilies based on overall body characteristics. So that I ended up in some fossils with 3 subfamilies or 2, or in one I am pretty sure it belongs to this Metopiny, the fossil L. Yes, so what I did then, I created a data set, a reference data set with extant species. I made that from illustrations from towns, and it's around with around 340 species. I placed landmarks and curves on 2 veins, and I also included 9 fossil species from the same formation. And then I just needed to check in the CVA analysis how well just this reference data set splits the groups. So that's the first result. Some groups, subfamilies, they split rather well, and some they are some intermediate between 2 others or mix completely. But here it's just shown the two first axes of the CVA. But they split quite nicely. And also, if we look at the shape differences, I won't go too much into detail, but you can really see some differences, and which I could use also later. And this is really interesting because it was thought that there's no information in wings in Darwin wasps to those different subfamilies. They all said, ah, well, there's 1 or 2 distinct wing shapes, but the rest is all about the same. And I think this proves it's not like that, and there's some information in there. What I did then is I compared the subfamilies with each other, and I calculated the procrustes distances and the differences between the subfamilies. So here in blue, I compared the 2 subfamilies that I needed to compare for the fossils, and they were above the mean difference within subfamilies. So I calculated the variation of wing venation shape within the subfamilies, and then I tried to see which ones were above. The above ones mean they are more distinct to each other than they are within themselves. But of course, I also have this example here where we have 2 subfamilies. They are very, very much alike. They are similar. The variation is very similar to the subfamily within. And I will show you just this example, this Triphonaini or Ctenopalmatini. It's this fossil here, which both subfamilies are based on the characteristics. They're the same. And the wing, I've tried to assign it just because of the forewing. And the CVA I added the fossil as a separate group and tried to check where it goes on the plot. And also, well, here there were just 2 axes. And the CV1 groups it to Triphonaini, to the brownish ones. The other one, here's a huge distance on CV2, but we should not forget that we compare extant species with a species that lived 55 million years ago. So I expect they're not to look the same. So I used a function called classify, which uses the CV scores. I did that on subfamily level. That's what you see here, and it groups it to Triphonaini. But when because Triphonaini, for example, they have taxa in it that really differ in the genus or other groups, tribes. I split them and I tested this on tribe genus level, and there they group to Ctenopalmatini. So in this example, those 2 subfamilies, it did not really work pretty much. But luckily it worked with many others. So for example, this fossil M, I had 3 subfamilies. The CVA, it's not in the group itself, but it's very close, and I have more CV score axes. It groups within Orthocentrini, the greenish ones, in tribe level, and also when we, and subfamily level, and also in tribe level. There it was in the Helictis group, which is also Orthocentrini. And it worked for many like this, but I also discovered something else. In this fossil L, where I was pretty sure where it belongs, but I tested it anyway with closely related or similar subfamilies. It did not group at all in the thought subfamily, because those 2, the greenish ones, they are the Metopini. I split them because the wings were really different in those 2, in the 1 genus, and it went to Ctenopalmatini. Great. In the subfamily. When I checked for a narrower taxonomic group, it went again to Ctenopalmatini and Campopelagini. then I thought, well, this is weird because the characters are very, very much metopiny.
You've got 5 minutes left.
So I checked, I had some discussion with Bernardo Santos, who's our collaborator with the UC dataset, and we talked, and it seems, at the moment, it seems like metopiny, they are very closely related to those Campoplegini and Ctenoplomatini, which could mean that this wing shape is this fossil belongs to some kind of stem group and could be an intermediate wing shape. And I'm now trying to calculate an ancestral shape and see if this is how it should look, or if I calculate this. So yes, I did that with some other fossils. And in the end, what does now a wing of a fossil Darwin Wasp tell us about the taxonomic placement? It pretty much depends on the analyzed subfamilies. So as I showed you in the first example, in some it does not work because they're very closely similarly looking, but it seems to be a useful tool for many others for taxonomic placement. And what I couldn't show you because of time issues, they give very good hints to shape variation within subfamilies, because there is also in tribes or genuses, which is quite interesting. And for the end, I want you to show something, a little excursion to not the sediment fossils, but to amber fossils. I supervised a little project with 2 bachelor students. I gave them this amber piece with a Darwin wasp inside. This is much younger, it's around 34 million years old. And they didn't know much about this fossil. I just told them that it belongs to around this group. And I could assign it to a subfamily, but they didn't. So they just took the wing and compared it with their dataset. And it was quite surprising how well it grouped with the right subfamily, the Fugadoentini, even though the fossil wing was not even flat. It was a little curved, but it worked very well in all of their analysis. So this seems kind of promising. Yes, and with that, here's a CT scan we did of this fossil. I would like to thank you all for listening, and to my supervisors at INA and everyone else that helped me in this project.